All right, so test our next time together of our first exam. Make sure when you come in for the exam that you sit every other seat. There will be about 25 more of you than what we've been getting every lecture, but plenty of time, plenty of room to spread out a little bit. Ideally, you're going to put your backpacks kind of off to the side just so they're not in your way. And try to get here a minute or two early so we can get situated and start right on time. I don't think you'll be super pressed for time. There's only 46 questions. So we got 50 minutes. We got more than a minute per question. You know, you probably won't have a ton of extra time if you check things over careful, but I don't think it'll be super. I can probably give a few minutes because there's not a class after this, and I don't have a class after this on Fridays. So I give you a little bit extra time if some of you need it. I just had the question, do I study things that are in homework questions that we didn't talk about in class for the exam? And I would say no. And I try not to have too many of those homework questions, just stuff in the text that we don't really talk about. There are a few just to kind of get you to look back in at the text a little bit. But I would say mostly study what we talked about in class. Look at those learning objectives. Look at those questions that I posted, the practice questions that I posted, just so you get an idea of the kinds of things that I asked. Also, don't forget, I know I've been kind of looking through the homework. I will go through and grade it all once it's due, and then I'm not going through it multiple times and rechecking things over and over. So I know there's still questions about, wait, I think I got this right and it's marked wrong. But I'm going to go through that this weekend and hopefully get all that taken care of. I noticed that it looked like not a lot of you had completed the section 7.6. Remember, right to begin with, we did that section on growth media. So don't forget that should show up there. We're going to start the rest of chapter 7 today. The rest of the chapter 7 is probably not made available yet, but it will be available Friday. So don't forget section 7.6. Also, we found a note taker, so don't worry about that anymore. Thanks for that person that came forward. Okay, where we left off last time. Again, had I planned just a little bit better or looked into my crystal ball a little bit more clearly, I might have included everything that we talked about in last lecture on Monday, but I didn't figure that quite right. But this chapter on eukaryotes, remember, will not be on Friday's exam. So just through cell structures, chapter 3. So last time we went ahead and moved forward into the chapter on eukaryotes and talked about protists. We started our discussion of protists, protozoa in particular. We had some particular structures that I highlighted, like the plasmalemma or the trophozoite stage in a protozoa's life cycle. And then kind of just for fun, I like to throw in kind of dinner table conversation stuff every once in a while. So this thing from Bill Gates's website about the most deadliest animals, we finished with that. When I post something like this, or when I show something in lecture and I don't post it, then it's something that you don't have to worry about, something that I just think is interesting. Okay, so we finished with that. Again, mosquitoes carry lots of viral infections in addition to the protozoa Plasmodium that causes malaria, things like West Nile and Zika and EEE. Remember that a few years ago, Eastern equine encephalitis. I had some deaths in Michigan from that. Dengue, there's like 14 or 15 different viral infections that mosquitoes carry. So if we just, I read that if we eradicated mosquitoes, then there wouldn't be any other animal that couldn't survive, like bats eat lots of other bugs besides mosquitoes. So, you know, something that they're kind of working towards, and engineering mosquitoes that don't reproduce, that sort of thing. But moving on into other protists, just going to spend just one slide here talking about algae. Algae are tremendously important in food chains, tremendously important in, you know, carbon recycling, you know, photosynthesizing, making oxygen, but they don't very often cause disease. If you get an overgrowth of algae, a red tide, and that happens all the time, Florida, for example, then yeah, that can cause some problems with humans, some respiratory problems, some fish die-offs, that sort of thing. But for the most part, they're not causing disease, so we don't really focus on them here. General characteristics, of course, they're eukaryotes, they're photosynthetic. Remember, we said that the protozoa are not photosynthetic. So when you talk about protists, they're either protozoa that are kind of animal-like, or algae that are plant-like. So when you Google that, whether protozoa are photosynthetic or not, you will get different answers, but OpenStax calls them not, and the algae are. The algae can be either unicellular, like here marine diatoms, those are unicellular. They can be multicellular, like red algae, which, remember, I don't know if I mentioned that specifically, but that's where we get agar, that solidifying agent that we use in media that comes from the cell walls of red algae. They can be colonial, like the Volvox, this kind of single celled that lives with other cells intimately. This giant kelp is a kind of brown algae that stretches for miles and miles in the oceans. So when you think about, well, you know, what's the difference between plants and algae? It's not just that plants are big and algae are small. It's a little bit more complicated than that. And just sort of in case you're interested, we didn't really go into the structures very deeply, but the vegetative structures on an algae all have the potential to become a gamete, okay, to have half the number of chromosomes, where in a plant, the reproductive structures, not all of them can become gametes. So if you ever wondered the difference between plant and algae, that's what it is. Cell wall composition, incredibly varied, so we won't even go into it. The diatoms, for instance, have silica in their cell walls, have some really cool looking shapes, but that's pretty much all I have to say on algae. Okay, so I want to continue that. Oh, I almost forgot. Plankton, like what is plankton? This is a word that you've heard of. Well, where does it fall into the scheme? It turns out plankton are defined by just, it's the taxa that's defined where they live. So microorganisms that live in a large body of water, that's what plankton are. So, and then they say, if you've ever heard the terms phytoplankton and zooplankton, phytoplankton are the photosynthetic plankton, and zooplankton are the ones that are not photosynthetic and just move around on their own. So I suppose SpongeBob's plankton, probably call him a zooplankton. He could be either a bacteria or an archaea or a protozoa, more likely. So we can narrow it down a little bit. All right, and then I spent a little bit more time on the fungi. Okay, so eukaryotic cells, of course, most often maybe think of fungi as being multicellular, so something that we're not really too interested in this course. But fungi can be more like mushrooms. They can also be mold-like, or they can be yeast. And of course, the molds in the yeast are the microscopic ones. So those are the ones that we are particularly interested in. I couldn't help show that picture on the right there of a puffball. Sometimes, especially when you go out in the woods in the fall, the conditions are just right. That like last fall, there was some huge ones that I saw. So kind of cool. The mushrooms have some of the same structures that we're going to talk about today. But primary characteristics of fungi, I'll just mention a couple. They are not photosynthetic. Okay, so remember, it wasn't until the late 60s that we really defined fungi as being different than plants, which is kind of surprising when plants photosynthesize and we consider fungi not to photosynthesize. And their cell walls are mostly made of chitin. There are some other possibilities there as well, but kind of the big one that's more recognizable is chitin. It's a polysaccharide, a little bit less complicated than peptidoglycan. Remember, all bacterial cell walls have peptidoglycan, and we don't see that in any of the eukaryotes or the archaea. Okay, so what's the significance? Why would we study fungi? Let's take some of your ideas. How would you encounter fungi in your life, or what difference does it make? What do we use it for? Think of any examples. Yeah. Ah, good. They are decomposers. Okay, nutrient recycling. All right, they are breaking down dead organic material. Good. What else? Something that you eat or drink, perhaps. Okay, it's yeast that are going to give us, rising bread, for example, alcohol, yeast that do the alcoholic fermentations. Other things like drugs, antibiotics. At least half the antibiotics that we have come from molds, like penicillium mold. Okay, so drug production. Kind of the other way, they can help us treat bacterial infections. They can also cause disease themselves. Some things that are more of a nuisance, like athlete's foot, are caused by the Candida fungus. Other things, potentially deadly infections, lung infections. We have a harder time treating fungal infections because they're caused by eukaryotic cells. Those are cells that are more similar to ours. So we have kind of less options of drugs to target the fungi and not our cells. Another thing that I want to mention here is that a lot of fungi can live in kind of a symbiotic relationship with other organisms. Okay, so symbiosis with other organisms. Of course, lichens are a symbiotic relationship between fungi and algae, usually. I was just reading, though, that they don't call that a true symbiosis because the algae actually grow better without the fungi. So anyway, it's kind of one of those gray areas. But when there's a particular important benefits that some fungi, when they live in relationship with the roots of plants, we call that mycorrhizae. Make sure I spelled that right. Oops, no H there, sorry. Mycorrhizae. Oh, I put it in the wrong place. Let me try, let me start again. Mycorrhizae, okay, so this is this is a symbiotic relationship. This is fungi that live in association with the roots of plants. And we think this is actually extremely common. Yeah, question. Ooh, is that what a truffle is? I don't think so. I think truffles, they mostly are underground. I would have to look up more. I don't know anything about truffles. But I'm glad, that was a good question. I'll have to look it up. These are fungi that live in association with the roots of plants and help the plants absorb water more efficiently. Okay, so help plants absorb water. So like I said, they're very common. I took this graphic, I think it was a lawn care website, where they were showing that their product helps those mycorrhizae interactions develop, and that makes the grass healthier. So it shows grass with that fungi and without. It's supposed to be, it's not a huge difference there in my mind, but apparently it's not, the grass isn't growing as robustly. So again, we think that's pretty common, and we think that probably, you know, helps lots of crops, again, absorb water better. All right, so I want to talk about some of the specialized names that we have for parts of fungi. And I've tried not to overburden you too much, but if we talk first about kind of the molds, or the fleshy fungi like mushrooms, they have some of these, again, same terminology that we use. They're always going to have some kind of hyphae. Okay, so that's the filaments. Something like a mushroom is going to have lots of hyphae underground. So when you see, I know like in my yard, every once in a while, there's a certain spot that all of a sudden mushrooms just pop up. And you think, how did they just grow so fast? Well, they have underground a huge network of hyphae. And then when the conditions are just right, like you get a lot of rain or something, there's a lot of stored energy in that hyphae and they grow very quickly. Okay, so either the molds or fleshy fungi have this filamentous growth, and we can consider those hyphae as either being septate or aseptate. Okay, septate meaning they have cross walls, and aseptate means without cross walls. So this picture here is not great. The drawing on the next page is maybe a little bit better, but here in the septate hyphae, you can kind of see these cross walls here. There's pores in those cross walls so that nutrients can move from cell to cell. And in the aseptate over here, you know, it's just the nutrients and everything, the nuclei are just kind of flowing freely between. So when there's no cross walls, these cells are actually multinucleate. And another name for that is senocytic. Okay, senocytic, that's a term that just means multinucleate. All right, those hyphae can either be vegetative or aerial then. A vegetative hyphae, the function is to, they function in absorbing nutrients. Okay, and if you have a big mass of vegetative hyphae, they're called mycelium. Okay, so an example, I'll show you examples in just a minute. Let me finish here. An aerial hyphae is considered reproductive. Okay, so you're going to have some kind of spore structure growing off an aerial hyphae. So the source of aerial spores. Okay, remember endospores for bacteria, kind of a dormant state, survival mechanism. Spores for fungi, reproductive. We're getting typically maybe hundreds of spores being released from some kind of aerial hyphae. All right, so let's see some other pictures of those things. Here's the ones from your open stacks. Again, this is a little bit more clear, those cross walls with the septate hyphae, and then the non-septate or cenocytic hyphae. We'll talk about that in a minute. Here's a picture of a fungal mycelium. So a mass of usually vegetative hyphae. This was from the old textbook. If you look at the units here, 3 millimeters, it wouldn't make sense for that to be a scanning electron micrograph. This is something that's visible to the naked eye. Some kind of light microscopy there. And then I like this, I grabbed this off the web, that just kind of helps get you familiar with some of these terms. Here's a hyphae growing out of a spore. Okay, so these spores are released from some aerial hyphae. Maybe they're carried in the wind, they land somewhere, and then they germinate. The hyphae grow out of them. Mycelium here, that's just a mass of hyphae interwoven. We'll get to that term in a minute. Here's some kind of aerial hyphae or an aerial spore structure growing off an aerial hyphae. So again, this particular one, we're not going to go too deeply. We're not going to talk about it all, reproduction of fungi, but this particular spore structure, when the wind hits it, they just disperse. Okay. All right. So that's kind of the basics of some of the filamentous fungi. There's just many other terms, and we could go into a lot more detail there, more than I want to. Fungi that have a yeast-like form. They're small, and their shape is often considered globular. They're not perfectly round like a cockeye, like a bacterial cell. They're just kind of globular shape. And I think the reason is because they often reproduce by budding. So that's kind of a asymmetrical distribution of the parent cell to the daughter cell. As we'll see shortly when we talk about bacteria growth, bacteria always, they get bigger and then they split right down the center, and the two daughter cells are exactly the same as the parent cell. So not so with yeast. With the budding, you get kind of just maybe a small portion kind of bud off from the parent cell. Okay. If that bud fails to detach, then we have another name for that structure, a pseudohyphae. Okay, so that's kind of what this picture is supposed to show over here. What's the advantage of that? Maybe that will help the yeast penetrate into some, really tiny crack a little bit more easily, if it can kind of, have almost like an appendage sticking out. Okay. Some fungi actually will exhibit both a yeast-like form morphology and a filamentous morphology. So we call that dimorphic growth. So exhibiting both yeast-like and filamentous growth. So here's a picture of some Candida fungi. You can see the kind of the roundest growth there, and the filaments both Typically, an organism's not going to be doing both kinds of growth at the same time. Typically, there's going to be some environmental cue that determines whether it's growing a more yeast-like way or a more filamentous way. If it's, causing, if it's in some kind of host and causing an infection, if it's in the yeast-like form, it's going to be able to disperse more easily in the bloodstream. In the filamentous form, then maybe it's a little bit better at invading tissues. But it's not always when it's here, it's always doing this, or when it's, doing this, it's always in this form. But it can go back and forth. Some fungi can. All right, and then really just lastly here, how to think about fungi, how they get their nutrition. We consider them chemoheterotrophic, and we're going to pick apart that term of how organisms get their nutrition when we get to our metabolism section. But a chemoheterotroph is going to get both their carbon and their energy sources from organic sources. So that that chemo, that refers to the energy source, that prefix, and the hetero, that means other, meaning it's getting its carbon source from somewhere else, other than not carbon dioxide. That would be an autotroph. That chemoheterotrophic nutritional acquisition is very common. We're considered chemoheterotrophs. Basically, it's organisms that eat stuff. So lots of bacteria, lots of organisms in general do that. Many fungi actually will get their nutrients from dead sources. So we call them saprobes, or saprophytic. Let me write that a little bit better. They're saprophytic. That means that they absorb nutrients from dead sources. Okay. So often you see fungi, on rotting logs, or if you see a tree with some shelf fungus on it, probably part of that tree is dead. Okay, it's very easy actually to absorb nutrients from dead sources because the cell walls are broken down, and that's not hard to do. Some fungi, though, are parasitic, and that means they get their nutrients from living organisms. If they are, they will typically have a modified hyphae called Astoria. So you can kind of see that here. Basically, their hyphae is just a little bit tougher so that it can penetrate cells that are still alive. Okay. And any fungi that does cause some kind of an infection, we say a fungal disease is called a mycosis. Okay, so that's just again our name for a infection caused by a fungi. So I think in the book they go in a little bit more heavily into reproduction. All fungi are going to reproduce asexually. So basically, the spores disperse from one fungi. There's no genetic recombination at all. Some fungi can also reproduce sexually, where the hyphal tips will fuse from one organism to another, and then again, the advantage there is you get some recombination, and maybe we'll get offspring that's a little bit better adapted for whatever environment it's in. And we often categorize fungi based on their reproductive structures. So there's a lot more terminology we could go into, but we'll leave that if you take another fungal course somewhere else. So, okay. Any questions about eukaryotes in general, whether it's protists or fungi, before we move on? Questions, comments? Okay. All right, so let's move on, kind of circle back around and hit the rest of chapter 7. I apologize. When I posted this, I hadn't edited this, and I just didn't look at it super carefully enough. So this introductory slide is a little bit wordy here. But we're going to start out first thinking about how microbes grow, and we referred to that a little bit. Maybe you talked about it a little bit if you're in labs, so you might see some things that are a little bit familiar. But I meant for these points to go up here in section 7.1. All right, so this is the only part of the course that requires a little bit of math, but don't get nervous about that. As we think about populations and how cells grow and how we can predict that, we're thinking about like growth curves and how that helps us predict the number of cells. We're going to think about counting cells and some different ways that we can do that in the lab. And then basically the rest of the chapter is thinking about what are the environmental conditions that cause bacterial cells in particular to grow really fast or maybe grow really slow. So things like the effects of temperature and pH and osmotic pressure, oxygen requirements. And hopefully you understand as we move forward here, especially that bacteria really can survive anywhere on Earth. So whatever niche there is, no matter how cold or acidic or whatever it is, there are bacteria that can survive there. I had a student tell me once that bacteria, we could get bacteria to grow from the sun. Probably not. Probably not fire. Probably anywhere else though. We could get some bacteria to grow or find that there were bacteria growing there. Okay, so as I mentioned a little bit, when we're talking about growth of microbes, we're not only thinking about how a single cell grows, but we're thinking about how they grow in numbers as well. But if you look at a single cell, bacteria are going to grow by binary fission. Okay, and that's a asexual, right? There's not any recombination going on there. It's just basically the cell gets longer or a cell gets a little bit bigger and it splits into two. So the cell enlarges and splits in two. Okay, so if you look at the bacilli shape, those bacilli will always elongate and a septum forms in the middle and then it splits into two. Okay, so those of you that are in lab this week and we're looking at maybe the E. coli that you gram stain, for example, you may have seen some variation, might have seen some cells that were really long and some that were really short. Well, the long ones were probably just getting ready to divide and the short ones maybe were newly divided. Okay. The time it takes for one bacteria cell to split into two, we call that generation time. Okay, so the time it takes for one cell to split into two. Okay, we also refer to that as doubling time. Makes sense. Okay, and that generation time is extremely variable. We have bacteria that under ideal conditions will split every 20 minutes. The mycobacterium, it might take them 24 hours to split. We talked about how that's the acid-fast genus that grows really slow because it has to produce that mycolic acid in its cell walls. Okay, so here's an example here over on the right. There's one cell, the chromosome replicates, a septum forms in the middle. When that septum is complete, okay, that's one generation. Say, for example, 30 minutes, okay? And then when each of those daughter cells splits again, now we have two cells that have become four. That's an additional 30 minutes, 60 minutes total. Okay, so on and so forth, where 2 cells have become 4 cells, now become 8, now become 16. Okay, so when the cells are doubling with each generation, we refer to that as exponential growth. So we'll talk about that a little more. This was the OpenStax graphic here. You can see probably a newly split bacterium here because it's still stuck together a little bit, so that septum probably just formed. The chromosome, remember, for most bacteria is circular, and so when it replicates itself, we get these rings then that will pull apart eventually, and we get 2 cells. So we'll talk much more about DNA replication in a few weeks. Okay. If we have a way to enumerate cells, which we'll talk about later, and we count the cells over time, we can construct a growth curve. So we can kind of mathematically kind of look at a representation of their numbers. So if we take say a culture in the lab and we inoculate it here at time 0, there's typically going to be a period where there's very little growth. Okay, and we call that the lag phase. Okay, so it takes time for those cells to kind of acclimate to the medium that they're in. They maybe need to make enzymes to break down certain nutrients that it's given. Again, very little or no increase in numbers during that lab phase. Once they get ramped up and are making the enzymes that they need, then it starts its fastest, it's exponential growth, where it's doubling its numbers every generation. So we call that the log phase. Cells in the log phase are typically of a very uniform size, shape, and antibiotics are generally most effective. Okay, so if we have a really rapidly growing population of bacteria, usually with most antibiotics, they're going to be most effective at killing the bacteria when they're rapidly growing. When they're not really growing, then most antibiotics are not as effective. And we saw that when we talked about how the cell wall grows and talked about the action of transpeptidases a couple times ago. But at some point, the nutrients start to become a little bit harder to find. Maybe the waste products build up. So we our cells will enter into a stationary phase where we have about the same number of cells dying as being reproduced. And then eventually the numbers that are dying will be more than the ones that are being produced. So we call that a death or decline phase. So this is assuming, this isn't taking straight numbers. This is probably on some logarithmic graph there. So I want to talk a little bit more about kind of representing our numbers on a graph. So first of all, let's understand logarithmic growth. So here, the species on the right here is growing logarithmically. That's another way to say exponentially, where they're doubling every generation. And you can see if we try to graph out numbers, just straight number of cells over time, that graph takes off upwards really quickly right away. So at some point, it very quickly becomes difficult to put that numbers high enough on that graph. So we have a way around that. We're going to chart them on semi-log paper. So I'll show you that in a minute. But contrast that if a population was just growing arithmetically. And that means that every time it reproduced, like it would add like a certain number. Like, okay, every year there was 2 added to the population, for example. Okay, then that it's gonna grow much more slowly. Now, normally in nature, we would, well, normally there are things in the environment that are going to limit the growth. So that's the only time that a species would grow arithmetically. Obviously, the moose population on Isle Royale, for example, that doesn't grow exponentially because the wolves are keeping it in check. Okay, so there's something in the environment, whether it's a predator or some nutrient availability that keeps it in check. And how quickly an organism grow, it depends on things like what species it is. Okay, so like I said, E. coli, it has a really fast generation time, just, I don't know, because it's E. coli, more so than some other species. So some species just reproduce more quickly. It also depends on the availability of nutrients. Actually, E. coli in the lab grows a lot more quickly than what it actually does in our large intestine. Okay, so there's, again, lots of nutrients in our large intestine, but apparently, we can make the conditions in the lab even more perfect. And also think about like other environmental parameters. So things like temperature, pH, things like that. E. coli will grow much more quickly at 37 than what it will at room temperature, for instance. Okay. So getting back to representing, growth, if we're graphing that out, like I said, this is similar to, on the left here, this is similar to this that I showed just a second ago. If we just graph this out, number of cells over time, after one generation, one cell becomes 2. Second generation, 2 becomes 4, 4 becomes 8, so on and so forth. 5 generations, it becomes 32 cells. So that's easy to graph to begin with, but then pretty quickly the numbers get really high. If we wanted to estimate, well, how many cells are there going to be after 12 hours, this would be very difficult to do. So if we graph instead on semi-log paper, where we have time on the x-axis and a logarithmic scale on the y, so 10 cells, 10 to the 1, remember this is 100, 10 to the 2, this is 1,000, or maybe it's 10 and 100. Anyway, when you plot it out on a logarithmic scale, if the bacteria is growing exponentially, you're going to have a straight line. So then it's very easy to say, well, let's see, how many will I have after 9 hours? And you can extrapolate over a little bit more easily if you've got a straight line. So sometimes that's helpful. Okay, let's see, we'll start here. We might not get all the way through actually doing some calculations, but let's start here and we'll pick up next week. If we want to, so that's one way to estimate bacterial numbers, is by graphing it out. And if we had time, we would do some growth curves in lab. It usually takes a good 4, 5, 6 hours to do a good growth curve, unfortunately, so I don't really have time. We can extrapolate mathematically, if you have some information, how many cells that you're going to have in a sample. Okay, so if we take, if the number of cells arising from a single cell is equal to 2 to the nth power, when n is the number of generations. Okay, so we could use this equation here, where we can, sometimes they have this represented as nf, meaning your final number of cells equals to your n initial number. This uses times 0, where n is the number of generations. So say you wanted to know if you start again, if your initial number is 0, your initial number is, you start with 1 cell. And let's see, after 1 generation, you're going to have 2 cells, right? That makes sense. If you start with 1 cell and after 2 generations, 1 times 4, you're going to have 4 cells. All right. After 3 generations, 2 times 2 times 2, okay, you get 8 cells. So you kind of get the idea there. So this, of course, normally we're working with really big numbers. Let's give you, so there's 2, we can use this formula, or we can also think about this in terms of just remember that your numbers double, right? So remember, you've got 1 cell to begin with here, to begin with. And then after 1 generation, it's going to double. You're going to have 2. After 2 generations, you're going to have 4. Okay, so sometimes you can just kind of double it to figure out a question like this. All right, so we'll do this together. There's a couple of questions in your homework to do as well. So let's work through this 2 different ways. Okay, if the question says, say you have 1.10 to the 5, that's 100,000 cells in a broth, and you know the generation time is 30 minutes, how many cells will there be after 2 hours? So if the generation time is 30 minutes and you have 2 hours, that's going to be 4 generations. Okay. So if we take our starting number of cells, let's just write it out this way. That's your starting number of cells, and you're going to multiply by 2 to the 4th. Okay, again, 30 plus 30 plus 30 plus 30, that makes 4 generations. Okay, that's going to be 100,000. So 2 to the 4th is 2 times 2 times 2 times 2, or 8. It's going to be times 16. Okay, so that's going to be 16 with 5 zeros, or 1.6 times 10 to the 6th cells. Okay. I would always give you a question where you did not need a calculator to do it, because I don't want to mess around with having any calculators in class. The other way that you could kind of just reason through this, okay, I said that you started out with 100,000 cells. That's your time 0. Okay, and the generation time is 30 minutes. Okay, so after 30 minutes, after 1 generation, this is going to double. Okay, so that's 1 generation. After a second, again, it's going to double again. That's your 2nd generation. Okay, doubles again. This is your 3rd generation. 120 minutes. Or say 1 hour. Okay, and then, yeah. Okay, so after your 4th generation, and did I do that right? 30, 60, 1, 2, oh yeah, 1.5 out, 1.5 hours, 2 hours. Okay, so you don't really need the equation. You can just, okay, it doubles. What commonly, sometimes the mistake that's made is that you count, you forget 100, students count 100,000 as the 1st generation instead of the time 0. Okay, so starting, know what you start with, and then when you double it the first time, that's your 1st generation. Okay, so again, when I make that homework, when I make that available after the exam, then you'll have some practice with that. And it talks about it in the book as well. So that's enough for today. I have office hours tomorrow. All Friday morning, I'll be in my office as well. So come by if you have any questions.